the whole genome system inside the human body. And actually making of this project very much many years, 13 years around it took, 13 years and what it took to come into this human genome project to get it completed. So about that we are going to listen completely in animated form. Yeah, you can start.
Human cell in books, we would have 3,300 books, each with 1,000 pages, where every page contains 1,000 letters. Yes, a human genome, which has about 3 multiplied by 10 to the power of 9 base pairs, can produce such an enormous amount of data. In fact, an ambitious international scientific research project called the Human Genome Project, or HGP, set out to do just that, to determine the complete sequence of base pairs of DNA inside the human genome. More precisely, the HGP aimed to identify approximately 20,000 to 25,000 genes in human DNA. The average gene consists of 3,000 bases. However, gene sizes vary greatly. Did you know that dystrophin is the largest known human gene, which has 2.4 million bases? Now, if you add the number of base pairs in the human genome, the sequences of more than 3 billion chemical base pairs would have to be determined under the HGP. Therefore, by aiming to understand the genetic makeup of the human species, the HGP served an important purpose. It was important because it is a sequence of bases in our DNA that determine our genetic information. 99.9% of all nucleotide bases are exactly the same in people. Yet we differ from each other, and that is because our DNA sequences differ in certain places. Scientists have identified about 1.4 million locations with single-base DNA differences, also known as SNPs, or single nucleotide polymorphism. Knowledge about variations in the human genome can equip us with more ways to diagnose, treat, or even prevent disorders and ailments that affect human beings. Similarly, knowledge of the repetitive sequences in DNA shed a lot of light on chromosome structure and evolution. Repetitive sequences, also known as microsatellites, are those stretches of DNA sequences that are repeated many times, sometimes 100 to 1,000 times, in the human genome. Also, they have no direct coding functions. In fact, less than 2% of genome codes for proteins and for more than half the discovered genes are still unknown. Advent of advanced genetic engineering methods, it was now possible to isolate and clone a piece of DNA to determine its sequence. So even though the HGP was a mammoth project, it was a viable one. Since the HGP aimed to store the entire genome information in databases, it required high-speed computational devices for data storage and retrieval. In addition, the HGP required improved tools for data analysis. Thus, the HGP is also credited with contributing to the rapid development of a new branch of biology called bioinformatics, which involves the application of statistics and computer sciences to the field of molecular biology. The HGP was a 13-year project that began in 1990 and was completed in 2003. It was coordinated by the US Department of Energy and the US National Institute of Health. Did you know that initially it was James D. Watson, one of the co-discoverers of the double helix structure of DNA, who headed the project? However, in the initial years of the HGP, Wellcome Trust of the UK became a major partner, though more contributions arrived from countries such as Japan, France, Germany and China, to name a few. Although the HGP had aimed to understand the genetic makeup of the human species, it also focused on several other non-human species such as the bacterium E. coli, yeast, a free-living non-pathogenic nematode called Caenorhabditis elegans, a fruit fly called Drosophila, and the laboratory mouse. Learning about non-human DNA sequences can lead to a further understanding of their natural capacities and traits, which may offer solutions in various sectors such as health, agriculture, and energy production. Apart from transferring information and technologies to other sectors, the HGP also intended to address ethical, legal and social issues, or ELSI, that may have developed while working on the project. Therefore, though the HGP was a mammoth and hugely beneficial project, it required enormous effort, cost and innovation, as well as caution to accomplish it successfully. In 1869, Friedrich Mies.
Okay, this is getting over with the topic of HGP. Now, Human Genome Project, as I told, there's a silent feature about it. 13 years it took, led by James Watson, as we know him before.
In the past, biological researchers could study only 1 or 2 genes at a time. This changed completely in 2003 with the completion of the Human Genome Project. With the entire gene sequence and advanced genetic engineering technologies at one's disposal, biological research could now be conducted on a larger scale. For example, biological researchers could now study all the transcripts in an organ, tissue, or tumor. It was also possible to study how thousands of genes and proteins work together inside our cells. HGP itself was made possible by 2 major methodologies of biological research: expressed sequence tags, or ESTs, or DNA sequence tags and sequence annotation. In EST or DNA sequence tag method, the focus was on identifying and isolating all genes that expressed as RNA. Actually, EST is the term for all genes that express as RNA. With EST, numerous databases of nucleotide sequences have been made available. It has facilitated the construction of the preliminary transcript map of a human genome. In the second methodology, the entire set of genome that comprised all coding and non-coding sequences was indiscriminately sequenced, and later different regions of the sequence were assigned with a function. The assigning of functions to sequences is called sequence annotation. For the purpose of sequencing, the complete DNA was extracted from a cell and divided into smaller fragments, since it is technically difficult to sequence longer fragments of DNA.
I'm trying to pause. It is not getting paused. No, it's not working the remote from here. Anyways, so identified the all those. Yeah, I told you about three types of RNA which are actually synthesized. So these the codings on DNA which codes for mRNA, that parts are ESTIs. Okay, so that they have that is one way they have removed. The other one way was they were, since the DNA is so long, some fragments of the DNA they used to uh, I mean molecular, what they will see is the scissors are the molecular scissors, endonucleases. With that they have cut and studied their pairing and what you call it as, to know about its genomes.
These DNA fragments were then cloned in suitable hosts such as bacteria or yeasts with the help of specialized plasmids or vectors.
This is biogenetics. Biogenetics, this is okay. Making of a plasmid. Okay, and this one plasmid is then put into the one E. coli, and then it is made multiplied and we get the desired material.
The
It's
vectors were called
biogenetics.
bacterial artificial chromosomes or BAC and yeast artificial chromosomes or YAC respectively.
Next chapter we'll be learning about this both. The most of it we'll be learning on CAC, sorry, BAC, that is bacterial artificial chromosomes. This is one of the plasmids just behind the what you see as a background. That is a artificially made plasmid taken from the E. coli, planting it back into the E. coli after making it with the desired. You saw the fragment they added it, and then they put it in the E. coli again and make them multiply, and we get the desired yield, whatever desired product, whatever we want.
The
This is what
cloning
when we do or make vectors of make bacteria as vectors. If we take yeast as vectors and implant all those uh, changed or modified genes in them, then we it is known as yeast, that is YAC, yeast artificial chromosomes. Okay, that is two ways we do the in the biogenetics, and that is why they're telling you that while doing this, vectors can be of two types. Whom we make the vector depends. That's why. Okay.
amplified each DNA sequence, which made it easier to sequence. Sequencing was then done by automated sequencers that worked on a principle developed by Frederick Sanger, who was also responsible for developing a method to sequence proteins and amino acids. As per Sanger's method, the cloned DNA sequences were arranged based on the overlapping regions present. For this, overlapping segments were generated. However, aligning this was not an easy task. As per Sanger's method, the cloned DNA sequences were arranged based on the overlapping regions present. For this, overlapping segments were generated. However, aligning this was not an easy task. Therefore, specialized computer-based programs were developed that annotated these sequences and assigned to a chromosome. Out of the 22 autosomes and 2 allosomes X and Y, chromosome 1 was the last that has been sequenced in May 2006. Did you know that chromosome 1 has the most number of genes? That is 2,968, whereas chromosome Y has the least number of genes, only 231. These challenging tasks would not have
Kindly
been
learn these digits. X
possible if
carries
advanced
most. X carries the
genetic
most. Y carries the least.
engineering
Yeah.
methodologies had not been put forth by the HGP. Consequently, paving the way for useful biological research. In the past, it was believed that proteins were the genetic material. However, of each
I'm not able to. Okay. Actually, human genome project is over. A different fingerprint than the other. Your siblings also, though the biological parents are same, still the siblings don't have the same fingerprints. Except identical twins, rest all have got, even identical twins, their fingerprints are different. Okay, so else uh, that is the way how, in forensics and all, it is used a lot to identify many things for some cases and all. Okay, so we are going to learn, like, how DNA fingerprinting is identified or done.
individual unique. The human genome has about 3 billion base pairs. Therefore, it would be a time-consuming and expensive task to find out the genetic difference between two individuals, as it would require comparing two sets of 3 billion base pairs. An easier and quicker solution to comparing DNA sequences is DNA fingerprinting, which is a test to classify and analyze DNA or the genetic information. In human beings, 99% of DNA base sequences are identical and are known as the bulk genomic DNA.
That's the first point. Bulk genomic uh, codes. 99% of it is the same. We all have 99% of the codes are meant for the same thing, like protein synthesis. All those things which is happening common in all of us is same. The process is same. So 99% of the information in each of us is same inside the DNA. Yeah.
The remaining 1% DNA base sequences differ and are present as a small stretch of repeated sequences known as repetitive DNA. DNA fingerprinting identifies the differences in this region.
Next is the next point to remember is repetitive DNA. Repetitive
To separate both genomic as well as repetitive DNA, the process of density gradient centrifugation is carried out.
Only 1% of DNA is actually separate.
Satellite DNA is lighter and bulk DNA is heavier. They get separated on the basis of their density. Graphical representation shows bulk genomic DNA
behind.
as a major peak.
We have to rewind a little only.
In this region.
Little more.
To separate both genomic as well as repetitive DNA, the remaining 1% DNA base sequences differ and are present as a small stretch of repeated sequences known as repetitive DNA. DNA fingerprinting identifies the differences in this region.
Which makes us
to separate both genomic as well as repetitive DNA, the process of density gradient centrifugation is carried out. As satellite DNA is lighter and bulk DNA is heavier, they get separated on the basis of their density. Graphical representation shows bulk genomic DNA as a major peak and repetitive DNA as smaller peak.
Stop now.
Known as satellite.
Okay, so by the centrifugation you have seen even the centrifugation when we went and to the we have gone to the university, right? In there we have seen the machine for centrifugation. So by centrifugating the DNA, we can separate because of the density differences. Bulk DNAs are actually bulk genomic DNA, which are common in all of us. Okay, 99% are actually heavy. Okay, while the other repetitive one are lighter in density wise when we centrifuge them. So we get the repetitive genomic DNA separated from the bulk, and actually we have to study only the that 1% we have to study, which is different in all the individuals. Okay, the bulk information is same. The bulk genomic DNA carries the same information in all of us. So we separated it. We don't have to waste our time in studying that. It carries the same information in all. What makes us different is that 1% of repetitive DNA. So see how they have removed it by centrifugation. Then, okay.
DNA. Satellite DNA is highly repetitive and consists of non-coding sequences. Based on the length of the segment, base composition, and number of repetitive units, satellite DNA can be classified as mini-satellite DNA and microsatellite DNA. Mini-satellite is a section of DNA which has a variable number of tandem repeats, or VNTR. It has sequences of 15 to 100 base pairs, repeated hundreds or thousands of times. On the other hand, microsatellite is a section of DNA which has tandem repeats of shorter sequences of 2 to 10 base pairs. Both mini-satellite and microsatellite DNA act as molecular markers in the DNA fingerprinting technique.
Okay. Okay, fine. This not able to stop. This satellite DNA means we are talking about that 1% repetitive DNA. Both have, but as you heard, in mini as well as microsatellite DNAs, both are having repetition of the codes. Mini has got larger coded things repeated many times, while micro has got smaller unit of codes which repeats a lot. So both of them together are known as satellite DNA because they are the one which make each other from individual to individual separate or different. Okay, and this both mini and micro satellites DNAs are needed for the DNA fingerprinting. So VNTRs, that is repetitive uh, sequences they're talking about. Yes.
Satellite DNA do not show any impact on reproducing ability and exhibit a high degree of DNA polymorphism or genetic variations within a population. DNA polymorphism is the result of mutations in either somatic cells or germ cells, which accumulates over generations. DNA polymorphism is the guiding principle behind genetic mapping and therefore helps in the DNA fingerprinting technique. The DNA fingerprinting technique was developed by Alec Jeffreys. This technique involved southern blot hybridization, which used radio-labeled VNTR as a probe. VNTR is a small sequence of DNA arranged tandemly in several copy numbers, which varies from one chromosome to another in an individual. A very high degree of polymorphism is seen in the number of these repeats, due to which the size of the VNTR varies from 0.1 to 20 kilobytes. Let's now study the steps involved in the DNA fingerprinting technique. First, the DNA is isolated and then digested by restriction endonucleases, which results in DNA fragmentation. Next, these DNA fragments are separated with the help of gel electrophoresis. and then transferred to synthetic membranes like nylon or nitrocellulose. This step is followed by the hybridization of the DNA fragments using a radio-labeled VNTR probe. Finally, the hybridized DNA fragments are detected by a technique called autoradiography, conducted using an X-ray film. Hybridization with the VNTR probe results in an autoradiogram, which produces several bands of different sizes. These bands provide a characteristic pattern to an individual's DNA and vary from one individual to another except in identical or monozygotic twins. Today, the accuracy of the DNA fingerprinting technique has further improved due to the advent of the polymerase chain reaction, or PCR, where multiple copies of a single DNA sequence can be made. DNA fingerprinting has also advanced due to the use of different types of DNA probes. DNA fingerprinting is used in many areas. It utilizes DNA as an identification tool and helps in forensic applications such as crime investigation. It also helps determine genetic and population diversity, forms the basis of paternity testing in case of parentage disputes, and also helps in the study of evolution and speciation. In this way, DNA fingerprinting has revolutionized the process of DNA testing, as it can identify the minute differences between various DNA samples. The human cell is an amazing structure.
These are the 1, 2, 4 steps. Okay, 4 steps in by which we identify the DNA. Now, what is the process? What they do? So the first thing is we're getting the sample. It can be hair, it can be blood from the, whose DNA has to be checked. Okay. Now, in this process, what they do first is, after isolating in a sense, taking the sample, whatever we are getting from, either from the hair or, as I said, from the blood or from the skin, whatever we get, they isolate it. And for isolation also, what they need is, using other examples, how they remove DNA. They remove DNA from, like, if I will put all RNA enzymes which digest RNAs, proteases, by using proteases, I can digest protein. All this kind of way, we add the enzymes. Now what we get concrete is DNA. We should not use DNases. Otherwise, that will also get digested. We don't want it to get digested. So, except that enzyme, rest all enzymes are added and separated from others. And now DNA, what they do is using restriction endonucleases. What is restriction endonucleases? We have learned very before. 2 types of enzymes present: endonucleases and exonucleases. They will cut the DNA parts. What we want, only that DNA part is taken into, because we know there are 2 types of DNA. Number 1, bulk one, and the other one is the bulk one, and the, what you call it, mini and microsat, that is a satellite, repetitive DNAs. And this is also isolated with the help of centrifugation. They do it. Okay. And after that, they use this endonucleases, and then we get the small fragments of DNA which we want to study further. This separation of DNA fragment, gel electrophoresis, after cutting it. Okay, see, after cutting it, how you will remove it? It is with the help of gel electrophoresis. We have